thank you very much for this opportunity to give me presenting my recent translational research, actually, uh, because the rest of my job is diagnostic one, and I enjoyed your recent talk about the disease causing mutation, which is a big issue now. This one is about the transcription factors. Today morning, there was a discussion in this regard in breast cancer, and mentioning breast cancer stem cells and its possible grand and great effect on metastasis. Um, SLUG and SOX9, as you know, both are transcription factor, major transcription factors, doing something important in breast cancer, and I present they cooperate together to regulate the good microenvironment and niche um, for breast cancer to be metastasized, actually. Metastasis is the major cause of cancer mortality. Metastasis is a multi-stage process, and we can consider a pathway, molecular pathway for that. And I would like to name it the metastatic molecular pathway. Successful initiation of metastatic growth, metastatic colonization, accomplished only by a minority of cancer cells that reach distant size. A small population of cancer stem cell is critical for metastatic colonization, and as you may remember, the seed and soil or metastatic tropism is the main dogma for this one. And cancer stem cell has some very well-known characteristic, including from self-renewal to apoptosis resistance, and about epithelial to mesenchymal transition. There's an emerging concept for metastasis in this regard, and it is the thing you can see in embryonic morphogenesis, in adult wound healing, and in metastasis. Wherever we have migration, invasiveness, apoptosis resistance, and these are all the characteristic of metastatic cells. EMT transcription factors are somehow known, including SLUG, snail, twist1, twist2, and the signaling pathway, which are already known, well known, are TGF beta and WNT beta catenin are in this category. There was a very good paper published in 2012 by Weinberg Group in New York. It says SLUG and SOX9 cooperatively determine the memory stem cell state as master transcription factors. SLUG and SOX9 master transcription factor, to have a very quick review on that. Actions of key transcription regulators is sufficient to convert differentiated epithelial cells into long-term repopulation epithelial stem cells. And as autoregulatory program, which is activated by SLUG and SOX9, involved in inducing and sustaining the stem cells and stemness. Adult stem cells are cancer stem cells similar to embryonic stem cells, maintain their stemness via master regulator mediated autoregulatory networks. And it was really nice for me. At the end of the discussion, I will ask to go back and address today morning discussion and see the expression pattern of these proteins, very limited number of protein transcription factors in the study of proposed a study by you, Doctor. Actually, the patient, it is well known and it is shown, confirmed that patients with primary tumors expressing high levels of both SLUG and SOX9 had a significantly lower overall survival rate compared to those non-expressed, this one in high level. Metastatic niche includes fertile microenvironment, supports stem cell maintenance and manages cell function and proliferation, including cell vascular network, soluble factor, nutrient, metabolic components, and ECM, as well as VEGF, membrane metalloproteases, LOX, and something like that. ECM and metastasis also address regulatory signals, sophisticated organization, and these two proteins, periostin and trypsin, as metastatic niche component for cancer stem cell. Why? Because in 2012, again in Nature, there was this published paper, Interactions between cancer stem cells and their niche govern metastatic colonization. Again in 2011 in Nature Medicine, it is published that breast cancer cell produce tyrosine C as a metastatic niche component to colonize the lungs. Okay, these two proteins, not transcription factors, periostin and tyrosine, are also important as metastatic niche in parallel to those SOX9 and SLUG, and they are studied at the same time, roughly in 2011-12, and published by different scientists and in different groups. We try to link between cancer stem cell and metastatic niche, addressing these two, and published this article in 2013. There is a crosstalk between breast cancer stem cell and metastatic niche as an emerging molecular metastatic pathway in tumor biology, and it is the thing that we hypothesize. As you can see, SLUG and SOX9 is at the top, and there are some intermediate components, then linking to tyrosine and periostin in normoxic and hypoxic area, and you can see the relationship they have with EMT and hypoxia and HIF1. In the same year, a paper published in Nature Cell Biology saying that perivascular niche regulate breast tumor dormancy, and it addresses periostin particularly and tyrosine also in the same pathway of TGF beta 1. In response to that, and considering our previous studies, we published an answer that perivascular niche governs an autoregulatory network to support breast cancer metastasis. The hypothesis was simply overexpression of SLUG and SOX9 transcription factors may increase the expression of periostin and tyrosine genes, and vice versa. Their knockdown may significantly underregulate and lower expression of periostin and tyrosine. I need to go faster. This study was done by my master student, and he recruited the constructs including SOX9 and SLUG, as well as silent RNA for SLUG and SOX9, and just transfected the cells via lentivirus, and then did the real-time PCR in MCF7 and MDA MB231 cells to compare these two invasive and non-invasive breast cancer cell lines. As you can see, in normal MDA and MCF7, there are significant different pattern of expression for SLUG, SOX9, periostin, and tenecin. It is the thing we've done in the lab by real-time PCR, and you can see MCF7, which is resistance to invasiveness and metastasis, showed a really lower, significantly lower expression of these four components. Talking about upregulation of the genes and transcription factors, the overexpression of SLUG and SOX9 significantly upregulated the expression of periostin and tenecin, which SOX9 is more obvious, and but as it is shown, their co-expression and cooperatively expression of these two transcription factor is much obvious, and it may propose that their co-expression and cooperation is significantly more powerful than single transfixion and overexpression. As you can see, there is about 1,000 times for upregulation of, in particular, periostin, and then about 50 times of tenecin for when they are co-expressed, compared to just single overexpression. Consistently, in knockdown SLUG and SOX9 downregulate periostin and tenecin. In this experiment, we've done in MDA cells, because it, as you could see, the expression levels of these components were high in MDA invasive cells, breast cancer cell line, and then we knocked down MDA 231, knocked down these SLUG and SOX9 in MDA 231, and quantitatively measured the gene expression of periostin and tenecin gene expressions. And as you can see, significantly both of them are decreased, and again periostin is more obvious and much more significant. To discuss, SLUG and SOX9 expression may have synergistic effect. SLUG and SOX9 co-expression is required for periostin and tenecin utmost expression. Periostin is likely one of the most important factor, even much more than tenecin, regulated by SLUG and SOX9. And as acknowledgement, I need to address my master student, which is now a PhD student in Spain, and the grant that it was funded by the university in Iran. Thank you very much.